It is my distinct pleasure to introduce our Path Presents speaker today, Dr. Vijg, and he's at the Einstein School of Medicine in New York City, where he's the chair. I forget what is it? The genetics? I think that's what it is. The Department of Genetics. Yeah. So and for those of us who are in aging and somatic genomics, he hardly needs any sort of introduction. He's made a huge number of contributions in that field over the years. I think he's published well over 200 papers. It's actually kind of frustrating when you go and you want to write a review or something like that, and you found out that he's just recently published one. So anyway, his research activity is really focused on molecular genetics of aging, and he's been doing some really cool stuff near and dear to my heart in somatic mutations, especially at single cell resolution. He's also dabbled in genome instability and transcriptional regulation in the intersection of aging and cancer. He was on I think a PO1 until very recently on that. He's also published stuff in molecular evolution and in personalized genetics or genomics. And so my hope with this invitation to give a talk, he's going to tell us about some of his really cool stuff that he's doing. So please take it away.
Thanks so much, Scott, for this very nice introduction. Really appreciate it. Okay, I guess everybody can see the slides. Just a little disclosure before I present our data and a little introduction to the topic, because as you could see from the title, I have an interest in aging and I have an interest in cancer and how they are related. And one of the mechanisms that certainly relates them or could relate them is somatic mutations. But let's first talk about aging, because most people know lots of things about cancer, but maybe not that much about aging, although taken into consideration the place where you are, and they have a great aging center, you probably know. Now, what you then certainly know is that I think one of the most impressive accomplishments of humanity through its maybe 200,000-300,000 years of history is actually this, what you see here on the left part. This is the life expectancy at birth, and you can see that in the 20th century, it went up very dramatically. You can see that here. In fact, this all started a little earlier, and it is the 19th century. It's really the 19th century that is the most magnificent century in human existence. All the major accomplishment, technological accomplishment that led to this and many other things were actually invented and applied in the 19th century. Now, the life expectancy increase that you see here is for France. We basically collected data for France. It was just because these data were accessible. France always has great data sets like other European countries like Sweden and so on. That's why you see these dips here. This is an enormous dip of the First World War. Of course, mostly males that died there. And here at the end, you see the great flu epidemic in 1918. And you see, of course, also the effect of the Second World War. But overall, there's increase, a little bit more, of course, for females than for males. Now, the reasons for this is, as I already pointed out, what happened in the 19th century. And that's when really there were some major scientific accomplishments and, of course, technological accomplishments. There was, of course, food security. It was the first time in human history that famines ended. It was no longer there. That was not only because of agricultural improvements, but also of improvements in food storage. Think of refrigeration, for example, that also came along in those days, but also transportation and other forms of storage of food. So with that problem out of the way, that was really a major reason for early mortality. And again, in probably virtually all of human existence, the life expectancy at birth was never more than 25 years or so, sometimes a little higher, sometimes a little lower even. But this was now immediately going to change because of all those improvements. The other one, other technology improvement, well, obviously the development of the germ theory, when people began to realize that you better use clean water, otherwise you have all these microbes that can infect you. And that was, of course, the major killer in those days. And people like Koch and Pasteur and Lister, they contributed enormously. They also realized that you should sterilize your equipment when you wanted to do some surgery or so. And then there was living conditions. Improvements in living conditions, getting away from all these crowding conditions, was probably a major factor in tuberculosis. Not so much antibiotics, that came later. Vaccination, of course, was always very important. Now, most of these improvements were really an enormous reduction in childhood mortality, like 200 deaths, childhood death on about 1,000 live births was normal in those days. But now it's less than 10 per 1 in 10 per live birth. So now, later in time, actually, we made improvements in keeping older people alive much better, and that you see here. Here you see the calendar age, calendar time, and you see here that there's an enormous improvement in the gain of years in very old people. You can see that here, 85 to 95, 100 years old, and there's a very dramatic increase. But you also see that it's actually, there are diminishing returns, and it's virtually zero now. So there are not many improvements anymore. And that suggests that there's an end to increases in lifespan. Again, remember, this is life expectancy, so it's not the maximum lifespan of the species. To calculate the maximum lifespan of the species is not easy, but in this recent aging outlook in nature, they sort of show the situation pretty nicely for a number of individual record holders. So you can see those that somebody who died in 1899 was 110, very rare in those days. But in 1997, Jean Calment died, and she was 122, and that's still the record oldest person who ever lived. After that, there was nobody who became any better than her. Now, I believe the oldest living individual is 119. If you look at the right, we plotted that here. So you can see here this, say, a record holder age when they died, and you can see the development, so it's going up and up and up, sort of what the same what you see here, essentially. But then after Jean Calment, this is Jean Calment, it's sort of got stuck, and there's a plateau. So there's, of course, fluctuation, but that's what it is. It doesn't increase any further. Now, the reason for this enormous increase here is obviously there's a huge number of much healthier people than ever before. There are millions and millions of people who live under almost optimal conditions. And that is why you see that they have a much higher chance of becoming a record holder. And that's why you see that. Now, improvements in conditions no longer help us now. We need to break through what is a ceiling, what is really the maximum lifespan of the species. Now, to put it in perspective, this line is the increase in centenarians, people over 100. This is the increase in supercentenaries, but there are not so many of those. So focus on the centenary. You see the increase in centenarians is almost exponential. So it's like an epidemic of centenarians. So there's an enormous push from centenarians, many still pretty healthy, that could all become record breakers. So at this time of Jean Calment, people expected that this would also go up. Maybe not as fast as the number of centenarians, but it would go up. It didn't. And when they compared the mortality rate of centenarians in this time, around this time, with now, there's no increase. So when you were a centenarian in the 1990s, your chances to die were exactly the same as they are now. So there's no improvement. That all fits the previous picture that I showed. Now, of course, there are two questions you can ask now. First, maybe, can we actually break through that barrier and do something that will allow us to fix aging and live longer, significantly longer, decades longer, maybe forever. We don't know. And the second is, of course, what causes this limit to life? What is the mechanism that holds us up? Now, first of all, and I think probably all of you know that aging is not a purposeful program. There's really no reason that aging is selected by evolution. At least if it is, we wouldn't know it. So I don't discuss it any further. Instead, a very likely cause of aging, a basic mechanism, molecular mechanism, was proposed by many others, but Vadim actually put it very nicely here as imperfectness at all levels, molecular, cellular, and physiological levels. And that gives you the fundamental mechanisms of aging. But there is a little bit of a problem there, because you can argue that, okay, if you can identify such a process, then we can actually maybe fix it, target it pharmacologically, and maybe do something about it. But that's actually very unlikely. I'm sorry, I have to be a little bit negative here. But this sort of goes back all the time to George Williams' paper in the 50s. And this is now pretty well known. Daniel, of course, is the expert there, and he will certainly improve me. But the argument here is that in normal populations, there will be an accumulation of germline variants that may have beneficial effects early on. That's why they were selected, but they can have adverse effects later in age. And there's no selection against that because it happens late in age. And there is a decline in the force of natural selection with age, of course, in the population. So, but that also implies that there are many of such genetic variants, and they must function across all organs and tissues and physiological processes. So eventually you always run in so many of these adverse effects that you really have to use enormous cocktails of drugs to do anything about it. So of course, this does not rule out the possibility that it is a sort of a driver of aging in terms of basic molecular processes that was always possibly always present during the evolution of life. We really cannot say that. Now, if there was such a process, a very early on, very highly conserved mechanism of aging, then that could be this. And that is where the somatic mutations are coming in. This was a paper from the 1950s by Gio Faya, a physicist, by the way, who began to realize that there are always somatic mutations. Of course, there are germline mutations as well, but somatic mutations also. And he realized that that could actually be a very good, very rational cause of the aging process, because it could also cause cancer. And cancer, of course, is very much associated with aging. There's an exponential increase in cancer risk with age. So in this paper, very nice paper, he actually made that argument. The problem, of course, that he had was that he didn't know much about what mutations actually were, but now we do. And I sort of made a schematic here about that. So somatic mutations, of course, well, germline mutations also, are really a consequence of erroneous repair or replication of DNA damage. And there's a lot of DNA damage. There may be 100,000 lesions like breaks or bulky adducts or crosslinks in a typical somatic cell per day. It's enormous. Now, virtually all of those are actually repaired. So it tells you already that DNA is not a very stable molecule, in contrast to what people think. It's actually unstable, and certainly under physiological conditions, but because we have an enormous repertoire of DNA repair systems, they keep it stable. And that's essentially the reason that it can be the main mechanism of transmitting genetic information, of course, DNA repair. But of course, sometimes there are errors. DNA repair is not perfect. And you'll get, those errors are called mutations, and that can vary from base substitutions to single base deletions or much larger deletions, maybe inversions. Chromosomal aberrations are also mutations, that is very large mutations. Now, of course, mutations are not useless. You will understand that if they do not happen, then there's no life on Earth, there's no diversity. So they need to be there in the germline. Most of it goes by the wayside in terms of death, they just die, these mutants. But the few that are successful, they are responsible for this enormous diversity of life and also perpetuation of life. So in a sense, it's sort of aesthetically satisfying, I would say, that mutations in the soma are also responsible for your death. This is life, this is death. Could make that argument. The problem that we always had since the 1950s was how to measure somatic mutations, because they are different, of course, from cell to cell. Germline mutations, yes, you simply sequence any tissue and then you got the germline karyotype or the germline genotype. But if you want to take a piece of somatic tissue and you ask for somatic mutations, they are different from cell to cell. So you can only really do that by looking into single cells, and I will come back to that. But we did know for quite a while that mutations do accumulate with age, somatic mutations it is. And this is just loss of the Y chromosome, in this case bone marrow, but you can also measure it in blood and I guess other tissues. There's a fairly early paper, and you can see that this loss of the Y chromosome is enormous. It's much bigger than we ever expected. There's a loss of other chromosomes as well, like the X, but it's much less dramatic than the Y. You may argue that it is because the Y is pretty useless, no? That may be true, but still, they measured the frequency of this in individuals, and they found that there's a clear association with mortality. So the more you have of this Y loss, there's increased mortality, and there's also diseases associated with diseases. Why would that be then? And possibly, I would believe it is because it's more like a surrogate marker for general genome instability or chromosomal instability. And that's probably why you see that association with mortality. But that's a little bit wishful thinking from my side, because it would suggest that somatic mutations are really important for functional loss during aging or for disease. And apart from cancer, we don't really know that. Now, also chromosomal abnormalities. If you heard somebody, a chromosomal person, already years and years ago talk about that, they all knew that when they look into the blood from a typical young individual, that about 1% of the cells actually contained at least one aberration. But if you looked in the blood cells of an old person, then you could see as many as 5%. So 5% of the blood cells contain at least one chromosomal aberration. And that's also true for other tissues, even the brain. So that's actually not little. I mean, this is a very big type of mutation, and then you find 5 of those. Sorry, you find one in 5% of the cells. That, I believe, is pretty dramatic. So we always knew, in a sense, that somatic mutations were pretty common and most likely not good for you. But we could never get access to all the other mutations, say the iceberg that underlie the tip of that, until we made our mouse models. We did a very simple thing, I think, making transgenic mice harboring a plasmid. In this case, we also made them for phage, but this plasmid containing a LAC reporter gene. And we could rescue that basically by excising it from total genomic DNA from any organ and turn it into the plasmid again, and then basically use the plasmid to transform E. coli. We modified the E. coli strain so that an active LAC gene would kill the cell. So only when it was inactivated by a mutation, it would grow into a colony. And then by relating that to, I'm not going into detail here, you can read it if you want to, it's a long time ago, you can actually calculate what the actual mutation frequency is at that particular time in the life of that mouse, and later we also made a fly. And we can also take the clone and sequence the plasmid and find out what the mutation is, either a point mutation, a small deletion, or a large deletion with one breakpoint here, and then the other, or here I should say, and the other breakpoint elsewhere. And we've done that quite extensively, and I can just summarize it briefly because it's already published a long time ago. This is the mouse. This is the mutation frequency, so the number of mutant LAC reporters over the total number recovered from that particular sample. And this is the calendar age of the mouse. This is the survival curve. So you can see that the mouse survival curve is pretty good. Although people would criticize it because they say, well, your mice didn't live under optimal conditions, so we can do much better. I believe that, but overall it's okay, I think. And you see that there's a pretty dramatic difference from organ to organ in terms of the accumulation of these mutations, with small intestine very high, liver and heart also reasonably high, and in our most important tissues like testis and brain, we see virtually nothing. Although I have to say, if you look at substructures in the brain, like hypothalamus, you see increased numbers of point mutations as well. Now, of course, we all were very happy with that, and we never dared to criticize our own work here, of course, but you do realize, I hope, that this was actually a bacterial gene, it's a LAC gene. It's completely DNA methylated, it's not active. And why would you think that that gene is representative for the rest of the genome? Probably it's not, although it might be, and actually it probably was to some extent, but still, what we want to do is to do the real thing and actually sequence the genome of all these cells from young and old people. And that we can do now. That's very, very recent. and I just summarize it here. There are roughly 3 types of assays. First of all, you can actually simply expand cells. You clone cells. You take cells from any tissue and you can bring them into culture and grow them and expand them into a clone. Then you sequence the clone, and the clone is, of course, representative for the original cell. So you don't need to do single cell sequencing, you can do clone sequencing. This is, I thought it was a very nice way of doing it, namely making organoids, which of course are clones derived from a stem cell. And they actually did that and found an increase in mutation frequency in these tissues with age. You can read that. It's already a while back. But nowadays, more popular is to take a tissue, in this case a liver, you fix it, you make slides, and then you make what they call a micro bulk. So you use LCM, for example, to take tiny pieces of it, and you do low input sequencing, whole genome sequencing, for example. And then you call variants. And when a variant has amplified into a clone, you cannot really, when there's one unique mutation in one cell, you cannot see them. But when that cell is grown into a clone, you capture the clone here and you sequence the clone. And that you can do. Those are still, of course, cells that are, this must have happened probably very early. And it's an expansion in vivo. An example of this is clonal hematopoiesis in blood. Now they find it for all human organs and tissues, but it was for the first time, I think, detected or found in hematopoietic stem cells. Now, what we believe is really the gold standard, of course, is true single cell analysis, and that was always prohibitively difficult because of the artifacts. When you do whole genome amplification of a single cell, there are artifacts of the whole genome sequencing. And this couple in my lab actually resolved that issue by designing a very reliable assay to do the wet lab work, so to say, and the computational analysis. What you see here is, of course, the next generation sequencing, the reference genome. When you do bulk sequencing, then this is a SNP, not is present in all these individual cells, compared to the reference sequence. These are sequencing errors, and this is your de novo somatic mutation, which you will of course never see. Now, if you sequence the single cell, then this unique mutation here will now amplify, and it's of course only present in one allele. So you see it as in half of the reads that contain that mutation. How do you know it's not a product of an error made during amplification? Well, this guy actually linked them up to the SNPs, and this is all linked, so it's true. This one is an artifact, and it's not linked. You can see that occasionally it's not. So we know that this is an artifact, and they're automatically rejected. Now, in practice, actually, when you have a good assay, she took care of that, then this is really not that much of a problem, much less than we always thought. Anyway, it works very well, and we can do single cell sequencing and detect base substitutions. Now, again, when this kind of assays came into play, and that's not that long ago, you saw a dramatic increase in the number of publications. When I looked at somatic mutations keyword and aging, British and American aging, then that's what you see. And you see there's almost nothing here in those days because we had no assays. And now you see the assay. So again, this is a very nice illustration of how important technology development is in our field. And I'm sure you all knew that already. Now, when you then take tissues, in our case, we used blood samples initially, because it's easy, not? From different subjects, different ages, then you can sequence the single cells. We took 4 single cells per individual, a couple of individuals. You can see this is cord blood. And we saw indeed what we expected to see, an increase in the number of mutations with chronological age. In cord blood, it's very low, a couple of hundred mutations, base substitutions per cell, but it goes up. And eventually, in centenarians, it can go up to 3,000–4,000 random base substitutions per cell. Sometimes there are outliers, and overall the variation is increasing. We have actually more individuals now which fit in the same pattern, but at that time, these were the only ones we have. It's very expensive. You still have to do a whole genome sequencing of every individual cell. Now, we did the same thing in human liver, but we could never get centenarians. You cannot convince them to do a liver biopsy, unfortunately. But so we depended on what our clinicians could give us. So it goes about 80 years old. You see that here. But the pattern is exactly the same. The only difference is the total number of mutations. at the same age level, when you compare it with the B lymphocytes, you see many, many more here. It's about twice the number, but in young age, it's about the same. Now, here we were a little bit hesitant because again, you know, you're still stuck with the artifact. You always want to check that, but you wanted to see something else as well. We had liver stem cells. We could also make organoids, of course, which Christina did, who did the work here. And she took those clones, those were clones, organoid clones from these livers. And she actually took the clones and sequenced the clones, so no need to do single cell. And she took single cells from these clones and sequenced the single cells as well. So now she could compare, and you can see that good for us, the results were exactly the same. So these single cells are very representative for an actual clone, it's the same. But when she now did not look at the stem cells, and remember, when you make an organoid, this is really a stem cell. So you look at stem cells here. When you look at the non-stem cells, at the differentiated hepatocyte, you can see two things. First, that the average mutation frequency is far higher. But second, there's an enormous variation from cell to cell. So that is the reason why you really want to do single cell sequencing and not so much take the clonal approach. Because when you take the clonal approach, first of all, you cannot analyze all organs and tissues. And second, you really capture what's happening in a progenitor cell or a stem cell. And that's not the same as in the differentiated cells. And we are mostly interested in those because they provide function. So if you want to make a link between mutation frequency and function during aging, then you better take the differentiated cells. And that's, of course, what we wanted to do. Now, I can show many more of these data, but I'm not going to do that. We actually put together a database, not only our data, of course, most of it is now from others. And this was done by Xixiang and Yuju. They put this database together, and you can read that it was published in Nucleic Acid Research, but you can go into the database and not only see all that data, but you can actually do experiments with it. There are all sorts of tools in it to look for mutation signatures and so on. Now, one other thing that I do want to show you briefly is, of course, when we were finding these fairly large numbers of mutations, and again, there's virtually complete consensus about it. Everybody finds thousands of mutations in cells or tissues from old people. We were wondering, I mean, we were aware of the results at the time already available from germline mutation frequencies. That was far lower. And now we wanted to compare that directly. And we did that here. This Brandon Milholland did that. He basically looked at germline mutation frequency in the same way as others did it, simply by comparing generations, looking at novel mutations in the offspring and compare it with the parents. And then, so he could quantify the number of de novo base substitutions. It's probably around 60 or so per cell in humans. And he did it for mice and he did it for humans. But then he now compared that directly in this case with the somatic cells using our new assay, then it's almost 2 orders of magnitude higher in the somatic. So obviously there's clearly a difference here. In some way, the germline genome is well protected, and maybe you're not that surprised about it. And as I told you earlier, stem cells are also lower than differentiated somatic cells. So clearly there is some built-in mechanism to protect what's really important, and maybe we can discuss a little bit later. Now, of course, the most important thing for us and for everybody, I guess, is there really a functional impact of that? Because you would say, well, somatic mutations, even if they're present in thousands of these mutations, how important are they functionally? Well, Maria Eriksson's group in Sweden, she was the one to look at these muscle satellite cells that you can clone. She took the clonal approach and looked at these stem cells by growing them into clones and then sequencing them. And she found something, also an increase with age, by the way, in the stem cells as well. But she also found that when there are more and more mutations, then she could see that the proliferative potential is lower in the cells with a very high number of mutations. So her conclusion was that there is a functional impact here, at least on the proliferative capability. And we started to look at that also a little bit more carefully in these B lymphocytes. And we subdivided the genome in whole genome and functional genome. Functional genome is the part of the genome that's really doing something in a B cell. So it's all the protein coding sequences that are actually transcribed in that cell, and the promoter regions, as well as the regulatory regions, such as enhancer sequences and so on. That's actually not little. In total, it's about 10% of the genome that's functional. And as you can see here, when you now look at the accumulation of mutations during aging, then this is far slower in those functional genomic regions than in the genome overall. And that, of course, suggests strongly that there has been a selection against that. Although there has been this selection that tells you that these must be functional, and mutations therefore must have a functional effect. It's also true that when you really look at cells in an old person, they really do have mutations in functional regions. You can read the paper and you will see that. Not many, but they clearly are there. So in other words, mutations that accumulate with aging can definitely still have a functional effect at very old age, in spite of the selection against it during the whole process that gives rise from stem cells to B lymphocytes. Now, some other evidence is a little bit more recent, also by the same couple who developed the single cell assay. They worked together in a collaboration with Vera Gorbinova, who has a beautiful collection of different rodents. They are, of course, they're rodents, so they're fairly closely related, but they're still very different in lifespan. And there's a tenfold difference between the shortest lived rodent, I believe that must be the mouse, as compared to the naked mole rat, which lives almost more than 30 years. It's about tenfold difference. So we looked at a number of these rodents and decided, well, what we can do now is we can take cells, any cell, and we took primary fibroblasts, and test if the cells from a longer lived rodent really have a better capacity to deal with DNA damage in terms of keeping the integrity, maintaining the integrity of your sequences, of your genome sequences. And we looked at mutations directly without doing anything, just spontaneous level. And what you see immediately here, it is the SNV frequency. You can see immediately that mouse is exceptional. The other species, guinea pig, blind mole rat, naked mole rat, human, which is the outgroup, is really, we couldn't see any significant differences, but the mouse, it is true, they are higher. We found that also, by the way, when we looked at these germline versus somatic mutations in mouse and human. The only difference between human and mouse was that mouse really did have a higher mutation frequency, and this is confirmed for other cells here. Now, more interesting was what happens now when you take these cells, you treat them with a mutagen. In this case, we took bleomycin, and you then wait for a little while, in this case 72 hours, and you take a look at the mutation frequency. Do we see that the same dose of bleomycin induces many more mutations in the mouse, short-lived, than in the naked mole rat or the human? And the answer is clearly yes, as you can see here. In the mouse, the bleomycin is doing something else as well. It kills the cells, because here we could only do 2 doses. If you do higher doses, then the cells are all dead and we couldn't measure them. But clearly you see a pretty dramatic increase in the mutation frequency. In the guinea pig, who lives about 3-fold longer, you see that also. You can see there's also a dose-dependent increase in the somatic mutation frequency, but it's clearly less than in the mouse. And in the blind mole rat, it's also there, but clearly less than in the guinea pig. In the naked mole rat, we really couldn't see anything, and the same is true for the humans. Now, one interesting finding, I think, was that that could explain this whole mouse thing. Why is it so dramatically high? The polymerase ETA is different. If you see that here in the phylogenetic tree, clearly they're separate here very early on. So maybe this is DNA polymerase ETA difference that's doing this in the mouse. That's a factor there. We don't know. But it cannot be the only reason, because as I told you, you see a difference here too. Now, interestingly, we just got this paper accepted, and then I got an email from Inigo Martin Corina from the group in Sanger in England, who basically did a little bit different approach, but essentially the same findings. They looked at not within rodents, but in different species you can see here, and they had multiple individuals of these different species, and they young and old individuals, and they could look at the rate of mutation accumulation during aging. And they saw an inverse relationship with the species lifespan. You can see here that too. So it's a different approach, but essentially the same results. So it's also clearly evidence, to some extent at least, that this mutation accumulation is really relevant, functionally speaking. But how could it work? And that, of course, we don't really know, because you really want to study that in much more detail, what happens actually. You see mutations accumulating randomly, and then they are getting into genes or not, or in regulatory sequences or not, and then what happens with them? Why does it reduce function? Now, this is like a summary actually to sort of to tell you what we know now about that. And the most important part is really the clonal expansion, because we know so much. So we know, for example, that when you look into typical human genetic disease genes, known diseases, and we have thousands of them, then, well, we always assume that they give you this disease because they are mutated in the germline. Yes, true, but now there's more and more evidence where we see that in this Mendelian inheritance of man genes, you see that also early mutations in the early embryo, which then expands, can actually give rise to these diseases also, maybe even more important than germline mutations as it now seems. We don't know that yet. So that is pretty clear. And you can have, of course, combinations of this, and those can actually have adverse effects on your tissues at some point. We don't really know. Certainly we know they can cause those diseases, but if they can cause aging, we don't really know yet. Nobody really studied it. Another factor that's also based on clonal expansion is somatic evolution. And I call this selfish mutation, not very original, I know, but the idea is that there's a fairly early mutation also during adult life maybe in a tissue, and when it is in a gene, and that is beneficial for the cell to grow out, it has an advantage here, growth advantage or a survival advantage, then it will expand as well. And that is what you see in clonal hematopoiesis, but you see it in all tissues. So that could, of course, definitely in some way or one way or another show aging aspects. Again, also this has not been really checked very in depth. In clonal hematopoiesis, this is going on now, but it's not easy to do. Now, the third mechanism, we know nothing about it. I call this mutational networking. I really don't exactly know why, but the whole idea is, of course, that when there is a random accumulation of mutations, how can it ever cause function? Well, any tissue, in every specific tissue, there are only particular gene regulatory networks active. They are the ones that give the cell its function. Not? So any mutation in any sequence participating in that network will either have no effect or it will have an adverse effect. And the more there are in these networks, the bigger the effect. So you would expect to see a gradual reduction of function. So it's very simple, not? I mean, in a liver cell, you don't expect that kidney function will go down, liver function will go down, or it will not go down, of course. And that's what you see. So if a sufficiently high number of cells will have slight deficiencies in function because of accumulating mutations, that could explain aging, not to some extent. So that's sort of the very hypothetical idea here. So the question is again, how would that work? What would you see actually? And the first thing you want to look at is the transcriptome, not? I mean, mutations, and you probably have an effect on the transcriptome. Now, a while back, we published a paper where we hypothesized that, well, one effect of mutations could be, and that was before I actually could look at mutations in such a great detail. I thought, well, then maybe when you have enough mutations, they will give rise to transcriptional changes at random. So some genes will then go up. I mean, most mutations, of course, are not in the gene, they're in regulatory sequences. So either the expression level will go up, it will go down, and this is random. So I would expect that when I look at the quantity of transcripts, particular transcripts, any transcript, this is a housekeeping gene, this is a housekeeping gene, this is a heart-specific gene, you would expect that at young age, all the cells are the same. They must function exactly the same. They're synchronized. So you don't expect any difference in expression level between these cells. But at older, we sort of hypothesize it would be, and there is. And actually it turned out in all genes that we looked at. This was way before we had all these fancy tools that we have now. So this was an endless checking and rechecking and controls and more controls to do that. But eventually we used real-time PCR. It's still very accurate. So that worked for us. Now, it took a while before this was confirmed, but now it's fully confirmed. There are lots of papers now who show increased transcriptional noise, as we called it, with AIDS. This is a very nice paper, I thought, by Steve Quake's group, who demonstrated that in human pancreas. And he also measured mutations there, by the way. So he sees that. So that could, of course, explain how increased mutational loads could cause aging, namely by randomizing gene expression. And that could, of course, give you all sorts of problems. And that's I show here. Again, this sort of summarizes what I just said. So you have these mutations like here. You see, this is one gene regulatory pathway. So here you have a mutation, here's a random mutation, here another one. They're all connected. So the more you get, then you get these slight problems in transcriptional output. And that's sort of summarized here. Now, what you would see then is, for example, that would affect the way a cell responds to a stimulus. Very important in physiological, in living, I would say. So when you do that, this is how it works for a young cell. But in an old cell, it's delayed. It happens later. Now think of the great heat wave in Europe for a couple of years ago in France. Who died? Well, older people died. They stepped out of their car, even a short distance from their air-conditioned car to their houses, and then they died because of they couldn't maintain their temperature so well. It's well known, by the way, that older people have problems with that. There are many of this kind of issues at old age, and those could be explained by this kind of mechanisms. But again, how to measure that correctly? We already noticed that we had really problems in accurately measuring this kind of transcriptional noise when we want to do it in a genome-wide manner. And then this nice paper came along, and they actually did this in a very different way. They looked at gene-to-gene transcriptional coordination, but the underlying idea is exactly the same. It's basically accumulation of mutations, or of course epimutations, that is causing this this variation in transcript levels from cell to cell. And they decided to sort of put together a tool, a computational tool that could measure that endpoint, that effect, transcriptional coordination. And it says it here, it doesn't measure transcriptional noise itself. It measures the dependency between expression levels of random subsets of genes when they have single cell RNA-seq data sets. And of course, there are many of those now. And what they actually found when they did this on all possible sets, and there are many of them now who found increased transcriptional noise with age, as I just told you, they actually find this, what they call GCL, is coordination level, goes down with age as transcription noise is going up. And in the data, when they analyzed Steve Quake's data, they saw that mutations, increased mutations in this particular tissue correlated with these reduced GCL values. I thought it was a very nice paper. I liked it a lot. Now, I'm not going to show you real reliable data. Well, I hope they're reliable, but I mean to say that this is more conceptual because we haven't done enough, our sample size is not high enough for this. So this is done by, I thought, very creative people, Ronnie Cutler, a student in the lab, Johanna Haidt, a postdoc, and Alex Maslov, one of my junior faculty. And they they irradiated cells, in this case, human lung fibroblasts. You can ignore this for a while. Well, you can ignore this. So they did irradiation, and then they took single cells and they isolated, they separated genomes from the supernatant when they lysed those cells. So they have RNA separately. They can do RNA-seq and DNA sequencing from the exact same single cell. Now, when they had to test it, obviously, and when they did that, they tested the genome analysis is simple. You just keep your cell nucleus, not. But this is always tricky. How would you know that you get the same RNA-seq levels? And they looked at this here. So they looked at what I call bulk single cell RNA-seq. So here they looked at the relationship between what they find in single cells and in the bulk. And you see it's a very high correlation. So we were happy to see that. So it means that the single cell RNA-seq seem to work very well. And now we have the bulk, the genome as well, so we can look at mutations. So you can see here the correlation coefficients. Now, so they then looked, of course, what they wanted to do was to look at the gene-gene coordination. And the idea was, well, if you look at young versus old, then we should see that. But they actually did the irradiation, which in a way is a surrogate for aging, not? And when you looked at the mutation frequency, they could see that in the irradiated cells, the base substitution level, SMV, is not higher. It's sort of the same. But the indels, insertions and deletions really increase, which is sort of what you would expect after radiation, I guess. And now they measure GCL, and they clearly see that it's actually decreasing with the irradiation dose. These are controls. These are known pathways where we know that the GCL must be maximized, and it is. And this is when you pick genes completely randomized, so you don't pick actual pathways or networks, and you can see that it is very low. So that's what you always included in these assays. And you can also see that when you look at the 6 single cells, each column is a single cell, and you look at the transcription levels, you can see that they vary completely randomly. This is, of course, what you would expect. Now, again, we are just started to do that in a way, and it's horribly difficult. I can already tell you that. Maybe somebody else can do it better. And so I'll show you sort of roughly where we want to go here. I mean, the baseline is, of course, that we want to look at individual mutations in the cell and then see what the transcriptional consequences are. That's really what we want to do. So here you can get SNV and indels, probably maybe other mutations at some point as well, profiles from single cells. We then annotate them to get these mutations to the nearest gene. It seems reasonable to assume that they would have an effect on the nearest gene. You can then do the integration with the RNA-seq, and then you can look at the expression levels. That's basically what you want to do. But you can see immediately a number of problems here. One of them is, of course, because you really look at, first of all, a fairly low number of cells, and the mutations are unique. So that's N=1, not? Anyway, I think that people like Daniel would be very useful here. At some point, I will probably call him up, but I do know that there are ways to approach this problem. So this is something for the future, but it gives you an indication where we want to go here. Now, I don't know how much time do we still have, Scott?
Looks to be, you probably have less than 10 minutes.
Oh, well, I can be very quickly. This is actually some work we did on cancer risk. And this is lung cancer. We worked with a clinician, Simon Vijg, who basically is doing this for a living, doing bronchial brushes to isolate primary lung cells. And we sort of worked with him to turn this into an essay for actually to look at single cell mutation spectra in nuclei, not in single cells. We did nuclei because sometimes you have frozen samples and then you can isolate nuclei, but not live cells. And you can basically see that we have this nice instrument that provides us with single nuclei. Now, the result when we do that in these single cells, here each dot is a single cell, and we do this for non-smokers. As a function of age, we find what we found for the B cells and for the liver, namely an increase in the SNVs, number of SNVs per cell, with age. That's what you would expect, not? And you find it. Also, this is also true for indels. It's always more difficult to measure indels, but you see that too. Now you can then determine the mean from the multiple cells per individual, so you can look at individuals. The same thing essentially, but now we look at the individuals. The donors, and they see the same thing. We see this increase with age. In this case, is just significant. So clearly, same what I already told you for B cells and liver. You can look at the mutational signature. I didn't explain this, but you can of course look at different mutation types and the neighboring bases, and then you can basically, there's a tool, computational tool, to actually find out what they call a signature, and you can then compare it with the signatures that were collected by Alexander for these tumors, and he has a whole collection of them. And since a tumor essentially is also a single cell, at the time that the tumor arose in a normal tissue, you can also look at aging. So we can basically compare these signatures with that, and you get what you expect. You basically see that a particular signature that is related to aging is also increased in our old subjects. Now, if you now look at the smokers, and this is only the mean per individual. So these are not single cells, but the mean of different single cells per individual. And you see very clearly that in the smokers, these cells not only have overall a higher mutation frequency, but also the rate with age is going up. And that's also true for indels. And now here, what we see here is actually, we see that the smoking signature is emerging. Also what you would expect here. Now, the most interesting result, and this paper should come out very soon, is actually this. Because we were interested, of course, is there a dose response? Some people smoke a lot, some people do not. So you want to make a link with smoking pack years, not? So the mutation frequency and the smoking pack years, and you see that relationship. These are all the non-smokers. This is an outlier. You have outliers occasionally. I'm not going to discuss this one, but you can see that initially this is going beautifully up, like the cancer risk is actually going up with smoking pack years. But cancer risk continues to go up for quite a while. Not here. Here you see that already after, say, 40 pack years or so, it basically becomes a plateau. Well, it actually even goes down, but this is, well, you can dispute that, but the reviewers didn't want us to say that it goes down significantly, but so we said that it doesn't go up. It reaches a plateau. Why is that? Why is this very high level smoker? Why not many more mutations? Most of them, as you can guess, had actually lung cancer, because otherwise they don't do the bronchoscopy. So, well, some of them maybe not, but there are other reasons, but mostly it's because of lung cancer. Now, of course, our hypothesis, as yours would be, I'm sure, is, well, their DNA repair system here is so good that they managed to keep their somatic mutation level low, and therefore they are survivors of lung cancer. In fact, only about 10% of all smokers, I believe, get actually lung cancer. So that could be one of the explanations there. Could, of course, also be that they are genetically better equipped to metabolize the mutagens in the tobacco smoke. That's another possibility. We don't know that. But certainly, this is actually what we believe is a possible mechanism that can help you to determine the risk that you run for cancer, for a variety of cancers, I would say. So the general conclusions now are that, yes, we can clearly see that somatic mutations do accumulate with age at a tissue-specific rate, also in the human. We show that in the mouse quite conclusively. Now, somatic mutation rate, it varies with species. We show that in the different rodent species. Also, environmental factors, smoking, for example, and stemness. So stem cells, mutation frequency is clearly lower, as it is for germ cells. Somatic mutation rate could now be a risk determinant for cancer. So if you're able to accurately determine the spontaneous somatic mutation frequency in cells, maybe buccal cells, maybe blood cells from smokers, you may be able to predict their cancer risk, probably for all cancers. We don't know that really. And we've shown that somatic mutations can have a fitness effect. The main question is still, can they contribute to aging in the form of giving your cells a functional deficit or causing diseases apart from cancer? That we don't know. So the most important thing we don't know. So this is the lab as it was. And Christina did most of the lab work with the liver cells, for example, together with Shishang, who's computational work. And then she is the one who did the lung cells. This is Lai and Xiao, who developed the assay. They are actually having independent faculty positions now in Minnesota. All right, thank you for your attention.
Oh, thank you. That was a super cool talk. This is an area that's near and dear to my heart, and I know for a fact that there are other people in the audience that are super interested in kind of the interaction between aging and cancer. So I will open this up for questions if anybody has any.
Maybe I should stop sharing, not?
Let's see here.
All right.
I have a question.
Yeah, sure.
Hi, I'm Rosana Risques, and I'm also working in somatic mutations, aging, and cancer. We use duplex sequencing, the method that Scott Kennedy invented, and others. And
yes,
we've seen a lot of what you've seen as well.
Yeah.
Mostly focus on TP53, which is a cancer driver gene, and so our focus comes from cancer interest. But we also see mutations in non-coding regions, and so we can see the age effect as well. My question relates to clonal expansion. So to what extent do you think the effect on aging is not as much transcriptional in terms of whatever mutations are producing in changes in the transcripts and the proteins, but the actual clones expanding and disrupting the tissue architecture.
Yeah. No, the straight answer is, I don't know that. So you could definitely, I think what you say makes a lot of sense, and it's very possible that it's only the clonal expansion that can do this. I gave the 3 possible mechanisms. 2 of them are clonal expansion. I'm just personally interested in what happens to a cell when it accumulates high levels of mutations. I had no time to show that, but we also try to pump many, many more mutations in the cell, not like a couple of thousand, but 30,000 or so, and then test what happens to these cells. So it's just basically my interest. It tests. I'm not. Yeah, of course I'm interested in the possible physiological effect on cell function as well. But I guess I'm a very basic scientist. But you're absolutely right. I think it's very well possible that it's only through clonal expansion. Good question. Oh, by the way, yes, that's a very interesting essay. We are also trying to set that up now, because you could already guess from what I said how extraordinarily expensive this is. You have to do multiple single cells, and when you do the single molecule assay, it's much easier, not? So we are trying to use that too. And one of the people in my lab, they made a couple of changes. At some point when you're interested, you should talk to us, and we can. Actually, it's in press now in Science Advances, the paper. We can get it to you if you want to. If you get me your email address, I will send to you a preprint.
Yes, please. Thank you.
Sure.
Of course, we are very interested.
Yeah.
Definitely. Right up my alley.
Yeah.
Anybody, any other questions here?
Yeah, if no one else has one. So one interesting thing might be to look at the ratio of synonymous to non-synonymous mutations over age. And if there is a functional effect here, you might see a change in that from young to older samples. Do you see that, or have you looked at that?
No, we don't. We do not see that. It's a good point. It's in our B cell paper. It's not. It's yeah. We also thought, of course, that we may see such thing. It's the same like there are all these kind of things like hotspots. Do we see hotspots? The answer is yes, we do, but not really dramatic hotspots. The hotspots that we see are mostly in B lymphocytes, the immunoglobulin genes, but for the rest, not a lot. So all these things we checked. It's all in the papers actually, but it was a little disappointing, to be entirely honest with you. So the only thing we see is there is this clear accumulation of mutations.
So here's a question. You know, you have that plateau in your smokers.
Yeah.
Right? But you don't see that in just normal aging. Just it keeps on. Looking at your data.
Good question also, because in the B cells you saw that we have centenarians there. So we were sort of hoping, as you know, you can hope when you analyze your data, that centenarians would be about the same, or maybe even lower than the 70 or 80 years group. But as you could see clearly, they're not. They're clearly higher. So mutations appear to accumulate all the time, irrespective of whether you're a centenarian or not. So we never reached a plateau in the age group.
Yeah, I mean, because it's like the cancer risk in the mutation mode.
Well, it's not exactly the same, because in the smoking group, they could. I mean, different individuals can of course have different somatic mutation rates. Not, we've never looked at it this far because simply the numbers. I mean, it's very expensive. You need to do longitudinal studies and whatever. We have not done all these things. But that could happen. But for lung cancer, which of course can appear much earlier, not in your 40s or 50s. So it's possible that you have very high lung somatic mutation frequencies already in your 40s and 50s, and then therefore you could have this high cancer risk. But in aging, you could argue, well, maybe you see very high mutation frequencies already at young age, and then you age faster. But we have never really seen anything like that. We've never seen like a person in his 40s or so to have the mutation rate or frequency of somebody in his 80s. We've never seen that. But again, we have never looked at progeroid syndromes. That is something like you guys have Werner syndrome. They have a whole collection. We've never ever looked at Werner cells. Here is something that you could do with your assay, not your single molecule assay. Have you must probably have done that, not? Rosanna, did you?
No, we haven't. I was just thinking about that. What data do we have? So the data that we have is actually for individuals with cancer. And what we see when we compare, so let's see, we compare individuals with cancer, TP53 again, but the non-coding and the coding. And so individuals with cancer, what they have is more clonal expansions, not necessarily more in the intronic areas. The rate of mutation seems to be similar, and it's the coding that are clonally expanded. That's what is higher.
Right.
So it comes to say, and that was part of the discussion that we are having, and I meant to ask that to you as well. So older individuals, it's not that the rate is different. So that's the other extreme, right? Like the ones that are healthy to the end, maybe the rate is the same. They are not clonally expanding. Somehow that process is being more controlled.
Yeah, no, it's possible, of course, because again, we haven't looked at that, but others have looked at tissues. So they've looked at clonally expanded mutations. I never really noticed a big difference. I mean, in our case, of course, we cannot say that there is a difference in mutation rates, so the rate of accumulation with age, but that is strongly suggested by the fact that we see this big increase in variation between individuals. Mm-hmm. So that suggests that there are these kind of rate differences. Mm-hmm. So, yeah, sorry, go on.
No, I do believe that it's an interplay of both. So definitely there could be an increased rate in many individuals for multiple reasons, right? Like decreasing DNA repair, for instance, right? That could be genetic even. Chemotherapy, any exposures, we've seen chemotherapy giving a higher rate overall. And so smoking, obviously. So I think that will happen, and there will be individuals, and it would be very interesting to see the progeroid syndromes, if those would also have an increased rate of mutation. But then I think in addition to that, there is the other layer, which is the clonal expansion. And so it
no, absolutely. I completely agree with that. That's really important.
Right. And so I think in individuals with cancer, we see both. Some of them, because of chemotherapy, for instance, have a higher mutation rate. But then overall, we see many more clones. So the clonal expansion of these precancerous mutations is happening as well. So
you know, what also would be very interesting is to look at these individuals who were treated for cancer early in life, and there's now quite a lot of evidence that they seem to show progeroid syndromes, accelerated aging later in life. Now, that is quite often explained because of the mutagenic effect, not of the cancer treatment. So you would sort of expect higher baseline levels of somatic mutations, maybe. It would be another population. You know, there's a lot of work to be done, actually. We should really do something together or so, because there's like a consortium grant, like program project or something. I see there are lots of opportunities here.
I could not agree more. We should definitely have a conversation about that. I just want to make sure there's nobody else has another question. I don't want to hog it with our interest. I just want to give anybody else a chance if they want to have a. All right. I think.
Oh, there's a chat. I think there's usually.
Is there a chat? No, I don't see anything in there.
Yes. Okay.
So anyway, well, I want to thank you again for giving that really cool talk. You know, this is just something that's an extreme interest to both Rosanna and myself. So we should absolutely.
Yeah. Let's do that.
Yeah, interact and maybe we can come up with something. Really
Thanks
cool.
a lot for the invitation. I really appreciate it.
Thank you again.
And hopefully we'll see live soon enough. Okay.
Yes, thank you.
Take care, guys.
Bye.